Good morning. My talk will be the genetic diversity or biodiversity, genetic diversity of Artemia in China. So I think the first part of my presentation will be a bit broader because I think I plan to give a brief introduction of our center, the Asian Regional Artemia Reference Center, and also our Artemia Sister Bank. Afterwards, I will leave the floor to our students to talk about the population genetic study results on the population from the Qinghai-Tibet Plateau, and then I will, at the end, I will propose some R&D idea. So, Asian Regional Artemia Reference Center, it was established in cooperation with FAO in 2016. So with the recommendation of the world Artemia experts, our center was established. I think a lot of experts in this room have witnessed the foundation and also the development of our center. So the center was officially opened in 2018. It's affiliated to Tianjin University of Science and Technology in China. So the center, what is the goal of the working goal of our center? It coordinates the multidisciplinary and interregional Artemia research. And also provides some technical consultation and services for the production of Artemia and their use. We are the member of IAAC and also the member of AAC, the Artemia Association of China. So this pictures shows you what we have been doing for the services to the industry. So we have, with the authority of by the Ministry of Agriculture, Rural Affairs and Agriculture, we have made two, national industrial standard for Artemia products. One is Artemia cysts, and the other one is frozen Artemia biomass. These standards, in fact, is good to guide the producer to make a good quality of the Artemia products. And also we involved in the revision of FAO technical paper menu on the production and use of live food for aquaculture, which was just launched at the end of May. So we are one of the revision party. We also translate some technical paper or technical information on Artemia into Chinese and to give it to more, guidance to the local farmers. We also do the technical training. So last year in July, we did quite successful training to the national level and to the farmers. So it's very, we had like around 100 participants. This year, we are preparing the second training course with the support of AAC, and we plan to do it in the hatchery in the south part of China, which is more, I think, we want to help the hatchery to let them more efficiently use of Artemia cysts during the macrobrachem production. So maybe this is just an example of our research at the center. So here we correlate fundamental Artemia biology research. So we are very ambitious because we really want to develop Artemia as a potential model organism. This is very important. I think not only us, I think we also want to collaborate with other scientists in the world. For example, Parisa, who is sitting there, she's also working very hard for that. So for this very small Artemia, only 1 centimeter. In fact, we managed to get a tissue from the brine of Artemia. So you can see that this is the what we call cerebral ganglion inside the brine of Artemia, and this is the neural cells of the Artemia. So how it looks like, different types, and also the ultrastructure picture of the synapse, so the antenna. And we also tried to study the regulation of neuroendocrine on the ontogenesis, so at the transcriptome level. And also we have already, set up a stable embryonic cell culture system, and this is very unique. In fact, for crustacean, there's no cell line available now for scientific research. But we try to make it of course, it's in the primary stage. We call it stable primary embryonic cell culture system. And with that system, we can do some research. For example, we already studied the infection progress of Vibrio on the embryonic cells of Artemia. So some of the information has been published in journal. And also we also studied the gonad development of Artemia. Just Gonzalo mentioned that the knockdown of nanos gene will make Artemia to produce more cysts than nuclei. But in this research, we also know that to silence a special gene called vitellogenin gene, VGR, it make Artemia produce less offspring. So yeah, all this kind of information, you can find it in Open Biology, which is published in 2013. And also, we also try to identify the hemocytes of Artemia. So Artemia has a quite diverse hemocytes, the type of hemocytes. So it has 7 types of hemocytes instead of 3, because normally for crustacean there are 3 types of hemocytes. So we have characterized all this this kind of these are the plasmocytes, and we use that information to study the response of these hemocytes on the challenge of nanoplastic. So, you know, nanoplastics, it's kind of pollutants in water, in freshwater, marine water, and also in brine system. So we studied that, and nowadays one of my PhD students who is from Bangladesh, and of course he is now busy in the lab and to study the impact of nanoplastics on the metabolomics and also the transcriptomes and proteomics using parthenogenetic Artemia as a model organisms. Okay, so we have done a lot of work, but I will not talk it in details. Now we have, in fact, Artemia sister bank or gene bank. Just now, Gonzalo mentioned that it's very important that to have the Artemia sister bank. In our center, we have a collection of more than 300 batches of Artemia cysts which are collected from over 50 locations all over the world. And the oldest sample in our center were sampled in early 1990s. Of course, all the samples are contributed by many people. Some of them are collected by ourselves, but most of them are collected by the scientists all over the world. I should mention that all these cysts are very clear. I mean, the information are very clear, and they are from which salt lakes, are they pure, and all this, we only collect information with very, very detailed information, and we are sure that they are the location where they are. And we also have database, including the biometrics, the hatching performance, the nutritional profile, and also genetic identity. We have the CO1 marker and also the SMP fingerprint of this data. Of course, to have this cyst bank is a kind of respiratory of our Artemia genetic resources, but also provide some materials for experimental research to guideline the utilization of Artemia in aquaculture. So this how is our database is, and recently by excellent work done by this gentleman, we managed to make a name card for our Artemia sample, our Artemia samples in the cyst bank. So you see, this is one of the name cards. So we put very the brief information here, the code at our cyst bank, and the collection site, the collection date, and the species information, and also molecular markers. For example, here is the SMP fingerprint code. We also put some phenotypic data, for example, the diameter of the cyst, the length of the instar-1 nuclei, and HUFA content, because especially the HUFA content, I think it's very important for Artemia to be used in the hatchery, and also the color of the cyst. And then we make like a QR code here. So by click this code, you will find more information. All this information are stocked in, are preserved in our lab. Okay, so diversity. China has quite rich Artemia diversity. We have 3 Artemia species and many Artemia populations. So just now, Gonzalo mentioned that we have Artemia sinica, we have Artemia tibetiana, and maybe the new one, Artemia sogolosi. But we have also one, we call it invasive Artemia species, it's Artemia franciscana. So it's not local in China, but they do now outcompete the local population in Bohai Bay saltworks. So we have like 4 now bisexual species and several parthenogenetica populations. In China, what is the situation of Artemia harvest? So annual harvest of Artemia cysts in China is now declining. So this figure shows you the row cyst harvest from 2019 to 2023. So we do have a declining of the harvest of Artemia cysts. On the other hand, China is the largest market demand for Artemia cysts. We annually consume about 1,000 to 1,500 metric tons of dry products, which account like about 50% of global production. Let's look at this picture. So, in fact, in 2003, we count all those Artemia cysts goes to what kind of species production. So about 75% of Artemia cysts are used for shrimp, Lithopenis vaname hatchery in China. And about 15% of Artemia cysts are used in the Macrobrochium, the hatchery, and a small amount of cysts, about 10%, are used for marine fish larvae production. of course, we don't have so many cysts available in China. So we are also the world's largest Artemia cyst importer. Annually, we import about 2,000 to 3,000 metric ton raw cysts. So here we mentioned about the raw cysts, the wet cysts, from Russia, from Kazakhstan, and from Uzbekistan. So we are rich in diversity. We have quite a lot of resources, but we also have very huge market demand for Artemia head cysts. Now I will go for the second part. So just Graham mentioned that in May of this year, we have received the requirement from FAO and to fill in the questionnaire of Aquaculture for the Artemia genetic stock in China. So just briefly introduce how did we work. So in fact, in the beginning, we are very confused for all this kind of the terminology, the words, and we are a bit worried how can we work on that. But after received the requirement, I think we have quite good communication with Graham and Daniela. We have the several online meetings, and we received like a kind of training. So we know the principle, the terminology, the concept, and afterwards we start working little by little. During the working, we also have some problems with the system, so we have feedback some things that we think it's not completed. Finally, we could manage to finish the task. So in fact, we got, how do I call it, the interface for user. So we got login information as a user. We can get in the system, and so we have imported 11 genetic stocks. Or we see that the Artemia population from 11 salt lakes in China. All these salt lakes are with commercial harvest for over last 10 years. This includes the all bisexual species in China, and also several parthenogenetic population. So the framework and information, I will not go in details because I think Graham just mentioned it's very clear. So just show you what kind of information we have input and how it looked like. So if we go to the assimilation, and we see that in China we have already, for crustacean, they are Penaeus venomys, is not our job, but we did the Artemia franciscana, sinica, Artemia tibetiana, and parthenogenetic population. Just show you for Artemia franciscana, if you just click in Artemia franciscana, these are the information that we have uploaded. So here is the reporting country, the name of the reporter, and also the species information. And here you see that is this species native to a country? No, so it's invasive. It's not native in China. Was its risk assessment done? We say yes, because when we search some literatures, there are indeed some literatures talking about the endangers of our native species, parthenogenetic population, because we know that Artemia franciscana has outcompeted this parthenogenetic population in the Bohai Bay saltworks. So all information like that. And also the details. So we have searched quite a lot of information. Unfortunately, most of the information are in Chinese, but we try to translate all this information and put all this information here. about the situation of Bohai Bay Artemia population. In fact, it's a mixed population of Artemia franciscana and local Parthenogenetica population. But yes, indeed, Artemia franciscana has outcompeted to our local species. So these are the species information. Then for Artemia, we don't have culture, aquaculture, so this part we leave it empty, but we'll go to the directly to the wild stock related information. And here, for example, we say, is there species also harvested from the wild population? Yes, so we give the data. For example, we gave a total harvest data of 2000 and last year, 2023, for this species, and also the total harvest value. So we think that Artemia cyst is the primary product of this species. So we put Artemia cyst as primary, and also there are also biomass production. So we put biomass, Artemia biomass, as a secondary product. Unfortunately, there is a problem with the system, so we cannot find the information here in the simulation, but we have put upload information in the system. And then we go to, we have also input information for the genetic stocks. So if you click here, you will have whole sets of information about Artemia franciscana in Bohai Bay saltworks. All this kind of information, is there genetic stock? What is the rationale for defining it as a genetic stock? So we put all information that we have. Some of the information are from our gene bank, but some of the information we measured during our work. So here we have, for example, the genetic stock. So we have information at molecular level, the CO1 marker, and also the SMP fingerprinting. We also entered the morphometric information of these genetic stocks. So you see a lot of data. And also the biological characteristics and the geographic distribution of these genetic stocks. We put the cyst diameters, the length of nuclei, Insta1 nuclei, and also the nutritional composition of these genetic stocks, and also the geographic location. Also for the taxonomic status of the genetic stock, it's a very complicated story. So you see that we have input all this kind of information here with a lot of supporting literatures. And also, is there the stock subjected to any conversion measure? We put information for the ex situ in vivo conservation, because these genetic stocks are preserved in our cyst bank. So here they are the code of cyst bank, here also mentioned. All this kind of information that after we upload it, and we can in this interface simulation, so you can find all information there. So I think we do finish just preliminary work, but I think we need to improve some of the data. So yeah. So now I will leave floor to my students, Liying, and he, together with Dr. Han, they are doing also a lot of work on population genetic study. Yeah.
I'm Liying. Okay. I'm the former master student at Artemia, Asian Regional Artemia Reference Center, major in population genetics of Artemia, supervised by Professor Liying and Dr. Han Xuekai. And now, I'm now doing my PhD in Jiangnan University, major in food development and multi-omics research of Artemia. I appreciate so much for the support of Ansel and Rose. And then I will report the progress of our works on population genetics, species distribution modeling, and the taxonomic status of Artemia in central Western China. Well, as the main organism in Salt Lake, the yield and the quality of Artemia cysts are influenced by environmental factors, human intervention, hydrological changes, which were mentioned in yesterday representation. For example, the RC is under threat of drawing up for decades. The salinity has reached 300 ppt, and the production dropped from 3,500 tons in 2020 to 1,500 tons in 2023. On the contrary, the salinity in Qixiangcuo, the largest Artemia production set on the plateau, has dropped to less than 40 ppt. So, therefore, studying genetic diversity and species distribution model in current and the future are crucial for conservation of Artemia resources. The genetic markers are used for population genetics, among which CO1 and ITS are conventional molecular markers and have been widely used in phylogenetic studies. But these two markers have short fragment and less variation that are insufficient to distinguish closely related species. But the SMP marker covering the whole genome with a large number and high repeatability and resolution can solve this problem. And there are more than 500 salt lakes in Tibet. I went to Tibet twice with my supervisors in 2023, and the environment is very special. Average altitude over 4,500 meters, a lack of oxygen and low temperature. Now, more and more salt lakes are getting fresh because of the glaciers melting and the high precipitation. In fact, it is very difficult to obtain the samples of Tibetan Artemia. Therefore, the studies are limited on population genetics. Since 2019, we have chances to investigate the Artemia resources in more than 10 salt lakes on the plateau and collected many cyst samples and available information. For example, when we use the CO1 markers to analyze the phylogenetics of Artemia from different salt lakes in Tibet, we can find that they didn't cluster together with the Artemia tibetiana. And we also read many literatures. And now, in our opinion, the problem for Artemia tibetiana is the complex taxonomic status. Are all populations belong to Artemia tibetiana? And did cryptic species exist? Meanwhile, whether human activity makes a difference because of artificial inoculation of Seneca in Dangqiongcuo, a salt lake in Tibet. So it is necessary to study the gene flow and the genetic structure among Artemia from different salt lakes in Tibet and the neighbor region. So we select 10 Artemia populations, 7 of them from Tibet, and we write down the question marker because we are not sure if all the population belong to Artemia tibetiana. And two populations from Xinjiang belong to Artemia parthenogenetic. One population from Shanxi belong to Artemia Seneca. We choose SMP to do this study. To our knowledge, it has never been used before. And then we got more than 40,000 SNPs by sequencing. First of all, we use Scrapped to calculate the genetic diversity in Linux. Overall, the diversity of Artemia in central western China was at a low level, but Tibet was higher than that in Xinjiang and Shanxi. Interestingly, within the Tibetan populations, genetic diversity tended to be higher in the west and lower in the east. For commercially exploit the salt lakes, the low genetic diversity implies that we must consider regional exploitation. Then we use the MegaX and IQ-tree to construct the phylogenetic trees in Linux and use a mixture to analyze the genetic structure. The results show that the 10 population could be grouped 4 major gene clusters. Apart from Xinjiang group and Shanxi group, the 7 population in Tibet were divided into Eastern group and the Western group. And we used GeneAlex to perform a mental test. We found that the significant positive correlation between the genetic and the geographic distance, indicating that geological isolation is an important factor for the differentiation of Artemia in central western China. Moreover, we found that weaker correlation among the Tibet populations, showing that other factors also affect the differentiation of Artemia in Tibet. For example, inoculation of exotic cysts or the bird migration. Furthermore, we use R language and the tree mix to calculate the variance and visualize the gene flow observed in the populations. Applying migration events equal 10, the model variance explained 99.9%. Therefore, this value was considering the input value for the model operation. And the tree mix revealed that Artemia sinica underwent unique decreational in gene flow in 4 salt lakes. These 4 salt lakes belong to eastern Tibet. And we can find the evidence because the inoculation occurred in 2015. And we can also find 2 strong gene flows. One is from Ivy Lake to Balikun Lake. The other is from Laguocuo to Dangqiongcuo. But we haven't found the evidence yet. And then we use Maxent, ArcGIS, and the ENM tools to obtain the potential distribution of Artemia in 5 different time periods. And the latter glacial period was 100,000 years ago. The glacial maximum was 20,000 years ago. Mid-Holocene was 5,000 years ago. The present and the future in 2070. The ecological Nikkei overlap between populations was small. This implies that the biological invention is relatively easy for Artemia. During the 3 historical periods, the fitness range of all groups, except for the Western Tibet, showed a gradual expansion. In the present, the Eastern Tibet region has the largest fitness area, whereas the West had the smallest. It should be noted that the range in Tibet will reduce rapidly in the future, with the range in the Western Tibet region contracting to almost 0 under 2070. Okay, the rest of my presentation is about cryptic species in Tibet. As we know, Artemia tibetiana from Laguocuo was named and recognized as a village species. The traditional view is that all populations from Tibet belong to Artemia tibetiana. As I have mentioned just now, from a genetic structure point of view, Tibet populations were grouped into Eastern and the Western group. Considering Tibet's unique environment and the presence a large number of salt lakes, it is necessary to conduct a comprehensive study on Tibet populations so as to further understand their genetic diversity and evolutionary status. Then we choose Laguocuo from Western Tibet, and the three populations Bangecuo, Dangqiongcuo, Qixiangcuo from Eastern Tibet. And the mitogenome for these four populations were sequenced and assembled. The results showed the Laguocuo is obviously different from other populations, such as the total base content, relative content usage, and so on. And the genetic distance and the phylogenetic analysis based on metagenomes show that Laguocuo has nearly different from the others, but closer to Artemia umiana. It is important to notice that the three populations joined with this Artemia tibetiana. This Artemia is from Yangna Pengcuo in Shuanghu County rather than Laguocuo. So these results suggest that there are maybe cryptic species in Tibet. Based on the mitogenome, we further developed the three mitochondrial genetic genes. Population genetic analysis based on three genes show that Laguocuo had the highest genetic diversity and significant genetic differentiation from the other populations. According to the haplotype evolutionary tree and the distribution, Laguocuo was far away from other populations, but close with Umiana. This is similar to the result based on the mitogenomes. To further confirm the difference and the taxonomic position between Laguocuo and the other populations, the morphological characterization of four Artemia populations were studied. The results showed the Laguocuo has the largest body size. In addition, the scanning electronic microscope observation revealed that the football knobs surface often occurs in cluster of two spines for Laguocuo males, rather than the cluster of three spines for other populations. Finally, Laguocuo and Qixiangcuo was selected for the reproduction solution experiment. The results showed there was an obvious hydrate breakdown and reproductive solution between Laguocuo and Qixiangcuo. We can see the reduced offspring number and survival, abnormal break stock between Laguocuo and Qixiangcuo. Above all, this study proved the existence of cryptic species of Artemia tibetiana by combining the result of molecular, morphological, and the reproductive solution experiment, considering the taxonomic states of Laguocuo, which was firstly named as Artemia tibetiana. The overall results indicate that the eastern Tibet populations, Qixiangcuo, Laguocuo, and Dangqiongcuo, could be cryptic species for Artemia tibetiana. That's all. Thank you.